Facilitated diffusion is a form of passive transport that occurs when large polar molecules are transported across a cell membrane, usually following a concentration gradient, through the use of transmembrane protein channels. Large polar molecules cannot pass through the phospholipid bilayer due to the lack of polarity along the hydrophobic lipid tails, of which will only allow small, non-polar molecules such as oxygen or carbon dioxide to pass. Examples of molecules transported in facilitated diffusion include, but are not limited to, polar molecules such as glucose and amino acids, large ions such as sodium and chloride ions, and large nonpolar molecules such as retinol. There are different types of transmembrane proteins such as carrier proteins, an example is integral glycoproteins, and channel proteins, an example being integral lipoproteins. A way to think about facilitated diffusion is like people traveling to a new country. The transmembrane proteins are like border control or customs when reaching a new country. The channel proteins are like tunnels used by particles within a cell. They do not bind to this structure, however, they provide an easy passage into the cell. Cell channel proteins can be thought as, for Americans, driving across the border to Canada. Instead of entering any buildings, you can just stay within your car, passing through. The carrier proteins, on the other hand, undergo conformational change in order to move a specific molecule across a cell membrane. This process is slower than the protein channels. They allow for specific molecules to be diffused in or out of the cell through the membrane. Using our travel analogy, it is comparable to the American flying to Europe. Here there are a lot more requirements in order to get through, and one must stay within customs. Customs can be seen as the binding particles to the channel protein. An example of a transmembrane protein is permease, and it allows for specific molecules to be diffused into or out of the cell. Permease binding is the first step in translocation. It requires molecular bonding between particles being moved in the transmembrane protein. A few ways this differs from simple diffusion is that facilitated diffusion requires molecular binding between particles and transmembrane proteins. It can be impacted by temperature, the rate of facilitated diffusion is not linear, or else simple diffusion is. Facilitated diffusion is always solute specific. Facilitated diffusion can be inhibited by an inhibitor, which binds to the carrier molecule. Facilitated diffusion can sometimes be considered an active transport if solute goes against the concentration gradient via a carrier molecule. A real-world example of facilitated diffusion is with glucose. Epithelial cells in the small intestine taking glucose by active transport after carbohydrates are digested. After this occurs, the molecules are released into the bloodstream and other parts of the body through means of facilitated diffusion. That's all on our video on facilitated diffusion. All the sources used are in the description below, along with the music. Thanks for watching.